bacteria. So answer was contribution in. Okay, so today we move into bacterial pathogenicity. So bacterial pathogenicity. Okay, so your learning outcomes. So there are a lot of terms today to be introduced. Okay, um, about the host pathogen interaction, how infection microbes are transmitted. Okay, some of these can be implied for viral or fungal infection as well, most of these. Okay, but the examples given will be for bacteria lah. Then explain pathogenesis, state the factors that contribute towards pathogenesis, mechanisms of pathogenicity and virulence factors that enable bacteria to invade and colonize. Then describe bacterial toxigenesis and differentiate between the two types of toxins. So when we look at if you look at bacteria bacteria, we have a lot of normal flora in our body, right? Normal flora in the body. Even our gut contains trillions of bacteria. But do they cause infection every day? No, it does not. Although some components of the fecal bacteria, the bacteria in our gut, they can be pathogenic. Okay, can be pathogenic, but not all will cause every day lah. Only when they breach the area where they are supposed to be, and they get transmitted into another area where they are not supposed to be. For example, from fecal getting into the urinary tract, for example, can cause urinary tract infection like E. coli. That's the cause of from the usually from the disease. Okay. So whereas some bacteria, they readily cause infection. Okay, you get transmitted from another host or an animal, or from fecal oral route also, or by touching contaminated objects. You are readily, it gives you an infection. Okay, it is a pathogen. Okay, so what makes this difference? Okay, so what makes some microbes infectious while others not? Okay, so firstly, what's a pathogen? A microorganism that is capable of causing disease. Okay, and non-pathogen that does not cause disease, and sometimes it's a normal flora, but not always. Some normal flora, as I said, can cause infection if they breach the barrier. Okay, sometimes during surgery, okay, a bacteria from the intestine can cause infect the bone and so on. Okay. Normal flora are microbes that colonize the human body and serve protective and nutritional function to the host. Okay, so they besides being there, not causing infection, it can also has protective effects for the host. As long as in their correct anatomic location, they will not cause infection. That is why not all gut bacteria can be probiotics. Probiotics have to be bacteria that they need to have certain criteria so that they do not cause an infection at any event. So only selected bacteria can be named as probiotics. Then we have opportunistic pathogens. Okay, opportunistic meaning they take the opportunity lah. They rarely cause disease, but in certain circumstances, like when a patient who is under medication, right? So they are immunosuppressed. This bacteria that normally can be evaded by a healthy immune system. Now it causes easily infects and causes infection. Okay, or in a like in the gut, when you take antibiotics, there are some pathogenic bacteria that are resistant mainly Clostridium difficile, they will they the good bacteria dies due to the antibiotic, and even the bacteria that you want to target, maybe another infection in the body, the respiratory or urinary tract infection, for example. If you are taking antibiotic for that, sorry. So if you're taking antibiotic for another infection, but you kill off your good bacteria, and some bacteria that are resistant to the antibiotic, they can proliferate and cause another infection. Okay, so these are opportunistic pathogens. Okay, so pathogenicity is the ability of an infectious agent to cause disease. Okay, and virulence is the quantitative measurement of pathogenicity. So one pathogenic bacteria can be more virulent than another bacteria. Okay, they can be of the same species also, but one may have certain virulence factors that makes it more pathogenic. So what contributes towards pathogenesis? First of all, it has to be transmitted from wherever, from the environment, from another person. Okay. Next, it has to be able to adhere to the cell surface, and then it invades, cause inflammation, and some can survive intracellularly, okay, without being killed by phagocytes, for example. Okay, and finally, some bacteria are able to produce toxin. So all these contribute towards pathogenesis. Okay, so let's look at transmission of bacterial infection. So first, they must gain entry into the body, right? So number one, through ingestion. Ingestion is by eating or drinking food or drinks that are contaminated. Okay, so these are the types of bacteria. Okay, so when we look at those specific lectures, we will come to this, but this is just an introduction. Next, through inhalation, okay, respiratory tract. Trauma and needle stick via skin. Via skin. So trauma meaning during accident, the skin gets breached, bacteria in the soil can enter skin and so on. Needle stick injury, arthropod bites, right? And also via sexual transmission. Okay, so some bacteria can be there in every root, okay, like Staphylococcus aureus is in several routes of entry. Some are more limited. Okay, so this is just a repetition lah. So these are the four routes of entry through respiratory tract, gastrointestinal tract, skin and soft tissue, and genitourinary tract. Okay, so they are generally can be transmitted from number one, from human to human through touch, okay, or sexual activity, via blood-borne, via blood contaminated blood that is transfused, or through IV drug use. Okay, indirect contact from the droplets in the air, fecal, oral, and fomite. Fomite are inanimate objects, objects that are non-living objects, which can be a mode of transmission, like sharing of towels, and children in the nursery, they share toys and so on. They can also transmit fomite through these fomite or inanimate objects. That is the indirect contact Next, vertical vertical is from mother to baby, across the placenta, during birth, or also via breast milk. some microbes can be transmitted. Next, from non-human to human, for example, soil. The soil contains a lot of, may contain spores, bacterial spores. We mentioned spores in our last lecture, kan? So it is through trauma, during accidents and so on, or when sometimes during gardening, some bacteria can be, or fungal even can be transmitted, okay, to the person from water, right, and from animal sources. For example, from the cat scratch disease, from insect vector, or even from animal excreta, right, urine or feces contamination, or from unpasteurized milk. Even milk that is not pasteurized is possible to be the mode of transmitting a foodborne infection. Okay, a few other terms. A reservoir is like what we call takungan kan. Okay, so it's the habitat in which the infectious agent normally lives. Okay, so I given an example here, the leptospirosis or apa penyakit kencing tikus, right? So here the reservoir is the rat, right? So that's the bacteria can grow and multiply inside the reservoir. Okay, and then from the urine of the rat, it can get transmitted to humans. Okay, so when an animal causes an infection, it can either be a reservoir or it can be the mode of transmission, a vector. Like a mosquito. We say mosquito-borne diseases, vector-borne diseases. Okay, so when infections spread between animals and humans, we call them zoonotic diseases or zoonosis. Okay, so what is an infection? It's a multiplication of an infectious agent within the body. Okay, so multiplication of the bacteria that are part of normal flora in the gastrointestinal tract or on the skin. Even on skin we have a normal flora. Okay, multiplication of these are not considered infection. Okay. And the infectious dose varies depending on their virulence factors. Sometimes a small amount of bacteria can cause infection, or sometimes you need large amount of bacteria to cause an infection. Okay, and in your community health, you probably would have, I'm sure you have come across all these terms. So some infection can be epidemic, can cause an epidemic. So it occurs more frequently than usual in a specific geographical area. Okay, have an unexpected increase suddenly in a certain geographical area, and then from there it can also become pandemic, or it can remain as an epidemic. Okay, pandemic, what we have all gone through, is a worldwide distribution declared when a disease growth is exponential, okay, worldwide. And then there is also what we call endemic, constantly present but at low level, the specific population. Like Malaysia, we are endemic for dengue, we have leptospirosis, malaria, right? Okay. Then some infections can also result in a latent state. Okay, latent state. For example, tuberculosis. Tuberculosis is caused by what bacteria? Mycobacterium. Mycobacterium. Tuberculosis, and it can be present as a latent state or active state, right? When it's latent, that means there is a small amount of bacteria still present. An infection that maybe happened in a person is young, and then the symptoms are gone, but the bacteria can be present in small amount, and then later it can become active. Okay, so we call that a latent state. And some infections lead to a chronic carrier state. Okay, carrier meaning the organism can continue to grow in the patient, in the person's body without showing any symptoms. Okay, but they can be the mode of causing infection in other people. Okay, so have you heard of typhoid Mary? Yes. Have you heard of typhoid Mary? Typhoid Mary is a chronic carrier of typhoid. So there's I hope your audio works here. Yeah, last week. So this is a short video showing. Is this? You don't have internet?
I do. Browse YouTube.
Oh, okay. Yeah. Okay.
In 1883, Mary Mallon left Ireland aged just 15 to seek her fortunes in
Yeah.
Yeah.
1883, Mary Mallon left Ireland aged just 15 to seek her fortunes in America. There, she quickly got to work as a cook for New York City's wealthiest families. In the summer of 1906, she was hired by Charles Henry Warren, a wealthy banker with a holiday home in Oyster Bay, Long Island. However, their holiday quickly turned south when 6 of the 11 members came down with typhoid fever. Victims of typhoid could suffer a fever, abdominal cramps, abdominal distension, intestinal hemorrhaging, and, in 10% of cases, death. The source of infection was water and food contaminated with excrement. Today, it's common knowledge to wash your hands after using the bathroom, but back then, that really wasn't the case. Immunization wouldn't roll out until 1911, and antibiotic treatment wouldn't become readily available until 1948. By 1907, in the New York area alone, 3,000 people have been diagnosed, which you would think would be a massive deal, but not yet, at least. It wasn't until the virus hit Oyster Bay, where it affected the affluent, that it became mainstream news. And so, George Soper was hired by the Warrens to investigate. His search quickly led him to Mary Mallon. Her unlikely partner in crime was peached ice cream. The cold dish merely froze the copious amounts of bacteria instead of burning them, which would have been the case in hot food. As the investigation continued, Soper began snooping around Mary's employment history. He discovered that of the 8 families she'd worked for, 7 of them came down with typhoid. Soper had a theory. Maybe she was the first ever documented healthy carrier of Salmonella typhi. He would need proof. Mary did not play ball, constantly denying Soper stool samples. He eventually needed the help of the New York City Health Department and even the NYPD. But don't worry, she wasn't arrested, just held on a desolate island in isolation for 2 years. North Brothers, to be exact. This was a bit of a moral quandary. There was an obligation to Mary's human rights, but also the obligation to the general public to keep them infection-free. People had been held in quarantine before, but only people who had clear signs of sickness. Mary appeared perfectly healthy. Compromises were struck up. Doctors suspected the bacteria were coming from her gallbladder and offered to release her if they removed it. Mary refused that deal, but did promise she'd never work as a cook again. 5 years later, she broke that promise and remained at large until 1915. She infected 25 people at the Sloan Maternity Hospital in Manhattan, infecting doctors, patients, and nurses, 2 of whom died. The NYPD took her back to North Brothers Island, again, where she'd spend the rest of her life. Upon Mary's death, some 20 years later, the doctors seized the opportunity to inspect Mary's gallbladder. And what did they find? Live typhoid bacteria living in her remains. The case of Typhoid Mary is a conundrum that still intrigues us today. Was she the villain, or was she the victim? Were the authorities right to violate one woman's individual liberties if it meant protecting the general population? And could they have done more to educate Mary on the dangers of what she was doing? If you want to see more content like this, please don't forget to like and subscribe, or leave a comment below.
Okay, that was about typhoid Mary. Mary, yeah. So she was from Ireland. There was a economic crisis where economic crisis where a lot of Irish people moved to US, right? And so this lady was later found to everywhere. She was a cook, she was a cook, and she caused food poisoning in a lot of the household. Okay, which later was found out to be due to Salmonella typhi. Do you know how it is transmitted? Yes? Fecal oral route. So, person who is a carrier like typhoid Mary, so you saw that it is, she carries it in her gallbladder. So, when you do gram negative bacteria or gastrointestinal tract infection later with Dr. Wan Shaida, you know the pathogenicity of it, how it gets there to the gallbladder, right? So she carries it, and it can during excretion, fecal excretion, when she goes to the toilet, it will be excreted in large amounts in the feces. So not washing hands properly and then preparing food can cause the um, bacteria to be transmitted to the people she's cooking for. Okay, and they said the popsicle, because it's a cold dish you don't cook, so the chances of not killing it is higher, right? So then they hired some detective to look into this case, and then so she was called Typhoid Mary lah, or Bloody Mary sometimes. Okay, so next courts. Let's look at court postulate. I think you have seen this with probably Lina's first lecture, right? So it's basically 4 criteria to give a causal relationship, right? How if there is an infection, is it caused by bacteria A, and does bacteria A can it cause the infection again? Okay, so around so these are the 4 criteria. Okay, so it must be isolated from every patient. The organism must be isolated from, if you say this organism is the cause of this disease, then it must be isolated from every patient who has that disease. And then if you grow it in pure culture in vitro, the organism must be able to be isolated. And then when you put it in an animal, it must infect the animal, and after you inoculate into the animal, you must be able to recover it from the inoculated animal. Okay, so that is like the gold standard lah. Although some of these criteria cannot be fulfilled, because some bacteria cannot grow in culture media, in vitro. They can only grow in animal, and then some bacteria or some microbes, they cannot grow in animal. Okay, but still this is generally the gold standard lah. Okay, for the causal if there is an infection. Okay, so these are some of the exceptions. So let's move on to virulence factors. So many of the mechanisms bacteria use to maintain their niche and the byproducts of bacterial growth can cause damage and problems for the human host. So these are very virulence factors that enhance the ability of bacteria to cause disease. So generally, for bacteria to cause disease, they need to invade the host and or produce toxin. Okay, so mainly these two. Okay, so to invade, first they must adhere to the bacterial cell surface, right? Okay, so how do they adhere? So these are the methods. Some of these we have already gone through during our first lecture, so I don't have to so pili. Okay, so these are examples of bacteria that have that Neisseria gonorrhoeae and Escherichia coli. So it can attach to the urinary tract, for example. Capsule, for example, Streptococcus pneumoniae, we saw transformation by the experiment by Griffith, kan? Where he showed that ability to produce capsule can be transferred between bacteria, even from dead bacteria to live bacteria. Remember? So that was the capsule, right? The ability to produce capsule in Streptococcus pneumoniae, right? And that interferes with phagocytosis. Glycocalyx, something like capsule, but they are more loosely. It's not a complex structure, not as complex as capsule lah. Okay, loose arrangement. Okay, so some bacteria like the Viridans group Streptococci, Staphylococcus epidermidis, so they have strong adherence to the endothelium of heart valves through these glycocalyx. Some bacteria have adhesins, okay, mainly on the pili, tip of the pili, okay, to help to bind even tightly on target tissue. Okay, next, biofilm. Have you heard of biofilm? No, but if you have seen pails of water or even your water bottle, if you don't wash it properly, you might feel some slimy. So that is biofilm. So biofilm happens when bacteria um, multiply. Okay, they multiply and grow and reach a certain number, what we call quorum. Quorum, you always say kan tak cukup quorum and all. So that comes from that quorum lah. Quorum, yeah, quorum sensing. So bacteria is biofilm formation is one of the virulence factors that is controlled by quorum sensing in bacteria. So when bacteria reaches a certain number in the population, it communicates between each other through cell signaling, okay, and it can turn on certain genes, for example, okay, certain signals, for example, to produce biofilm. Okay, so the ability of producing biofilm it's to the bacteria's advantage lah. Okay, so it can get more nutrients, it can access into the organs more easily because it's a slime layer kan, and then it can avoid phagocytosis and so on. Okay, so biofilm is especially a nuisance when it forms on foreign bodies, because they can, like you see in pails, plastic pails, plastic water bottles. So they can also adhere to prosthetic joints, prosthetic heart valves and intravenous catheters, and cause a new infection in a patient who's probably on a catheter for quite some time. Such when the biofilm forms, it can cause an infection at the site or where the catheter is inserted, or where the prosthetic joint or heart valves are placed. Okay, so this is one of the many factors that cause nosocomial infections. You've heard of nosocomial infections, infection that occur in kat mana? Hospitals, right? Nosocomial infections are infections that occur in the hospital. Patient goes to the hospital for a different disease, maybe infection or non-infectious, other forms of non-communicable diseases, and then acquire a new infection because of from the hospital. Okay, so one of it is the biofilm causing it. Okay, another can be because of antibiotic resistance. A lot of the hospital environment can be a source of antibiotic resistant microorganism. Even the doctor serving the patient, that's why it's important to wash hands in between patients, right? Because you don't want to contaminate your patient with a new infection from another patient, or even from the surrounding, like touching the beddings and so on. Okay, and it can also form on native structures like on the heart valves. And in cystic fibrosis patients also it's a bigger problem because they have this genetic disorder, isn't it, to produce more sticky these excretions from sweat and mucus, so it is more vulnerable to be um, to have for bacteria to form biofilm there. Okay, and the dental plug is an example of biofilm. So this image here, if you can see, this actually two tooth gigi in and the gum here, if you can see clearly. So this one scraping from this area here, looked under the um, electron microscopy, can show you an example of a plaque biofilm. So biofilm can be formed by different types of bacteria or a single bacteria. Okay, so this one maybe the teeth usually will have multiple colonies of bacteria. This is another example of biofilm formation by Staphylococcus aureus on an indwelling catheter. So this is electron microscopy, so you can see the Staph aureus here and the biofilm here, the slimy matter. Okay, so that was adherence through papilli, adhesine, biofilm, and so on. And then invasion. Some bacteria have enzymes that they can secrete to help them invade. Okay, for example, hyaluronidase and collagenase. Okay, so this degrades the hyaluronic acid and collagen to help the bacteria spread through the subcutaneous tissue. Okay, and then hemolysins and leukocidins. So these are enzymes that can lyse erythrocytes, red blood cells, and also white blood cells respectively. And the examples, coagulase. Okay, some bacteria produce coagulase enzyme. So this accelerates formation of a fibrin clot using the fibrinogen from the host. They accelerate formation of a fibrin clot so that the fibrin clot that they form can protect them from phagocytosis. So Staph aureus is one example of a bacteria that is able to produce coagulase. So this coagulase in the lab is also used to identify Staph aureus. Okay, and fibrinolysin is the opposite of it. So Staph aureus has both. Okay, it can produce coagulase, it can also produce fibrinolysin. Okay, after it can also produce fibrinolysin to lyse the fibrin clot. Okay, that it had formed, and to allow it to spread to the next cell and so on. Okay, so these are all invasive enzymes. Immunoglobulin A protease. So this degrades IgA on mucosal surfaces, allowing adherence to mucosal membranes. So some of these bacterial examples are given here. Okay, and then virulence factors that interfere with host defense mechanisms like our capsule. We have mentioned that many times, so it's important for you to remember. So another example besides Streptococcus pneumonia is Neisseria meningitidis. Okay, Neisseria meningitidis. And then cell wall proteins of some gram-positive bacteria like the M protein or in strepiogenes, which is antiphagocytic. Protein A of Staph aureus. Okay, this binds to IgG, preventing complement activation. So these are also proteins that allow them to interfere from the host immune activity. Okay, immune response. Okay, and the ability to survive within phagocyte. Okay, for example, mycobacterium. So some bacteria are able to survive within the phagocytic activity and even survive within the cell, live within the cell. Okay, so here it inhibits fusion of phagosome with lysosome. So if you look at this diagram here, this is what happens in a normal healthy phagocyte, right? So C3b, what is C3b? It's a complement opsonin, right? It's an opsonin, so it aids in phagocytosis. So if you look here, so the bacteria, the C3b adheres to the bacteria, and then the phagocyte has these C3b receptors. So it engulfs the bacteria and forms the phagosome, right? Phagosome. So the phagosome now inside the phagocyte, and the phagocyte has lysosome. So lysosome has all these enzymes and funny chemicals that will kill the lice and kill the bacteria inside the phagosome by forming the lysosome, right? So bacteria kill the lice and then this remove lah from the cell. But what mycobacterium can do, as you can see here, so this is what happens in the right side here on the blue. The bacteria is blue here. The bacteria is mutant. Mutant for PKNG. So this is okay. So this is what happens in normal healthy, like the previous example here. This full diagram here. So what happens is bacteria goes in to forms the phagosome. Phagosome fuses with lysosome and bacteria can be killed. But in pathogenic mycobacteria, they have protein kinase G. They have this protein kinase G to block the phagosome lysosome fusion. So this fusion here does not occur. Okay, so when this doesn't occur, so bacteria remains inside the phagosome. So here it doesn't fuse with the lysosome. So this is an ability of Mycobacterium tuberculosis lah, okay, to survive within phagocytes. Okay, so makes it difficult to treat or difficult to combat pathogen. Okay, so we have seen adherence and invasion. So finally, toxin production. When I say toxin, I hope when I say finally, I hope some people will wake up, but still tak bangun. Okay, so toxin production is the third mechanism. Not all bacteria can produce toxins, but if you can tell me which type of, there are two types of toxin, generally, which one is the one we saw in our first lecture? Endotoxin, which is present in
the wall, the cell wall.
Yes, gram negative bacteria. So do all gram negative bacteria have endotoxin? Yes,
Yeah.
LPS, lipopolysaccharide, if you remember, right? So gram negative bacteria has endotoxin. The other toxin is exotoxin. So exotoxin is produced by both gram positive and gram negative bacteria. Not all, okay. So depending lah on the species. Okay, so when you say endo meaning?
Inside.
What's the meaning of endo?
Inside.
Inside kan, dalam. Exo means
Outside.
outside, but then endotoxin is outside the cell. So how will you remember this? So it's actually part of the bacteria. Okay, it's an outer cell, part of the outer cell membrane, but it's still part of the bacteria. Okay, endotoxin. Exotoxin is secreted toxin. Okay, it's released by the bacteria. Okay, so endotoxin is a cell wall component of the gram-negative bacteria. It is weakly antigenic. So as we look into endotoxin, exotoxin, I hope you can think of the difference between these two, because it's important to know the differences between endo and exotoxin. So if it is weakly antigenic, so it has poor induction of protective antibodies, right? Meaning that it cannot be used in production of vaccine. You know, for vaccine production, you use for bacteria, you use parts of bacteria, right? Not the whole bacteria. So Sometimes you use the whole bacteria as well, like in Salmonella typhi, it's an oral live vaccine which is attenuated, so it's not pathogenic, but it has components. But most vaccines, we use parts of the bacterial structure which is highly antigenic, meaning that it can produce a good immune response, so that the next time if you are infected with the actual bacteria, your body has the immune memory cells to produce the antibodies for it. But endotoxin is weakly antigenic, so tak sesuai lah. It's not suitable to make into vaccine. Okay. So it's released into host circulation following lysis or during multiplication. So you see the three components of an endotoxin of the glycopolysaccharide is the lipid A, the core polysaccharide, and the O antigen. Okay, the toxic part is the lipid A. Okay, the lipid A is the toxic component. Okay, so this is just to remind you again about how gram-negative bacteria has this LPS or endotoxin in the outer membrane. Okay, so if you endotoxin, LPS, must remember gram-negative bacteria only. Okay, so how they act? Okay, they cause septic shock, which is characterized by fever, hypotension, and DIC, disseminated intravascular coagulation. So this is caused by clotting, blood clots in the capillaries, which can cause tissue death. So mainly the reaction response caused by endotoxin, the septic shock, is mainly due to the release of all these inflammatory cytokines from the host immune cells, okay, which causes all these. Then here you have TNF, anti-mononuclear necrosis factor, so it causes capillary permeability increase, right? From firstly, the LPS itself causes increase in nitric oxide, causes vasodilation, and so on. Okay, causing hypotension. Okay, so these are the three characteristics of endotoxin to the host, right? Okay, so this is another chart from your textbook showing that. Whereas toxin, exotoxin, they are secreted polypeptides. Endotoxins are lipopolysaccharides. Exotoxins are polypeptides, secreted, not part of the bacteria, but they are secreted. Even when the bacteria is no more there, but the exotoxin alone can cause food poisoning, for example. Not infection because no bacteria that cannot infect, but the toxin itself can cause. For example, if the bacteria has been killed with the cooking process, but the exotoxin is heat stable. If the exotoxin is heat stable, it can still remain and cause food poisoning. Okay. So what are exotoxins? They are produced by several gram-positive and gram-negative bacteria. They are secreted polypeptides. So between polypeptides and polysaccharides, protein and polysaccharide, which one is more antigenic? Polypeptide, right? Polypeptide, you've seen that in your immunology. Okay. The genes are usually located on plasmids or on bacteriophages. Okay, so they can be transmitted from one virulent bacteria to another non-virulent bacteria, making it more virulent, right? The genetic component. Okay, and these exotoxins, they can be treated with formaldehyde, acid, or heat to convert the polypeptides into toxoids. Okay, and these toxoids can be used in vaccine production. Okay, one example of a toxoid vaccine is the DTP. DTP covers for what? Diphtheria, right? Tetanus and pertussis. Remember the recent case Elena also shared, pertussis. It's highly likely that they were not vaccinated lah, the children. Maybe lah, okay, or was not effective, the vaccination. I don't know, that was not, I don't think they anything about that, I'm not sure. Okay, so vaccination can reduce the vaccination can protect kids from pertussis, whooping cough, right? So the way it was formed is through production of toxoids from the bacteria. Okay, so toxoid are inactivated form lah of this exotoxin. Okay. And how bacteria release them through a secretion system. The most important is type 3. So they have a structure called injectosome. They can inject this exotoxin directly into the host cell, okay, so as to protect itself from the immune related cytokines and so on in the extracellular area. Okay, so that's how they inject it in. So action of exotoxins. Many exotoxins have an AB subunit structure. Okay, so A for activity lah, so that A subunit possesses the toxic activity. B for binding. The B subunit responsible for binding to specific receptors on the cell membrane, host cell membrane. So a lot of several important exotoxins acts by ADP ribosylation, and then they can, for example here, this is diphtheria toxin. Diphtheria toxin is also has an AB subunit. So we are talking about exotoxin here, not endo. Exo is a secreted toxin. And diphtheria is a gram-positive bacteria, Corynebacterium diphtheria. Okay, so this toxin, when it gets into the host cell, it can it acts on the EF2 elongation factor 2, causing the toxic ADP ribosylates host protein elongation factor, stopping protein synthesis. Okay, so causing cell death. So that's how it works on the host cell. So a lot of these exotoxins, they have this AB subunit. Okay, another example is the cholera toxin. So when you look at gram-positive bacteria, we will see how this works further. Okay, so cholera toxin. Cholera is a gram-negative bacteria. Okay, so I just put an example of gram-positive and gram-negative bacteria. Also has the AB, but here it acts on the GS molecule here. Okay, and it can stimulate. This is the so it gets into the intestinal barrier, intestinal lumen, and it can increase AC, annihilate cyclase, right, and which increases CAMP. CAMP causes what? Reduced uptake of sodium and chloride, causing a release of water, so causing this watery diarrhea. Okay, so that is the act of the toxin. So cholera toxin is what causes the watery diarrhea in cholera patients. So this is how it acts. Okay, the B for B binds to the cell surface and gets in and does what it does Okay, botulinum toxin is another toxin. This is a transmitter. So it acts on at the neuromuscular junction. I think we will look at it when we do gram positive bacteria. But have you heard of botulinum toxin?
Yes.
How? What is it used for? Yes,
Basically,
Botox. Botox. So that is human manipulating the bacteria lah for
cosmetic.
cosmetic as well as some medical users for people who have this stiff neck. Sometimes they can't turn. It's turned on one side, right? They use small amounts of this toxin to relax the muscles there. Okay, and also the eyelid. Some people have this. They cannot control their blinking, right? So it can be used. You think they are doing it kenip mata kan? They blink their eyes, but actually it's because they cannot control the muscle function. Okay, so that for these cases, small amounts of Botox it can be used to control that lah, so that it relaxes the muscle there, contracts the muscle there. Okay, Botox for cosmetic purpose also works in that same way lah. People who have this what you call crow's feet, right? Crow's feet, when you smile you can see your muscle here, you get wrinkles kan. As you age it becomes like permanently there, right? Because of repetitive use. So some people like to get rid of that with Botox. Okay, so sometimes you don't know whether they're smiling or not. Okay, so Botox is from this botulinum toxin. So in the patient, botulinum toxin can cause paralysis. We look at that during when we do gram positive bacteria lah. Okay, so these are a list of toxins, exotoxins produced by gram positive bacteria, and these are by gram negative bacteria. Okay, some examples. So don't be shocked to see these. You as you go through the lectures, you will get more used to these. Okay, so we have diphtheria toxin, we saw earlier, tetanus, botulinum, Clostridium difficile. Difficile is a diarrhea causing bacteria in the gut, right? The one that is resistant to many antibiotics. So when your gut bacteria, good bacteria dies because you take antibiotics for some other cause, maybe, right? This Clostridium difficile can start proliferating. Okay, in people who have it in small amount in their gut, you can start proliferating, producing these toxins and causing diarrhea. Okay, they call it antibiotic induced diarrhea. Then you have multiple toxins from perfringens and toxic shock syndrome toxins. Staph aureus is a very important pathogen. Okay, so you can see, you always will see Staph aureus everywhere, every system. Erythrogenic toxin in Staph pyogenes. And then gram negative bacteria, they have endotoxin, right? But they can also have exotoxin. Okay, some can also secrete an additional toxin. Okay, and some are heat stable and some are not. Okay, so this is our last learning outcome. So what's the difference between exotoxin and endotoxin, if you can recall from what we saw today?
Endotoxin is from gram negative.
Yes, endo is part of the bacteria, exo is secreted. Okay, for endotoxin, the bacteria must be present to cause infection. For exotoxin, bacteria, the toxin alone is sufficient to cause
infection,
food poisoning and disease. What else? One is only present in gram negative. Okay, the other is can be both. Apa lagi? The component? Polypeptide. Which one is polypeptide?
Exotoxin.
Exo or endo?
Exo.
Exo is a polypeptide. Endotoxin is a polysaccharide. What else? What else? The location of genes. Exotoxin is on plasmid or bacteriophage, right? So it can be transmitted to like Corynebacteria, Corynebacterium diphtheriae. There can be non-pathogenic diphtheriae, and there can be those that are transfected by bacteriophage carrying this gene can be toxigenic. Okay, whereas endotoxin is in the bacterial chromosome lah. Okay, it's part of the structure kan. Toxicity, exotoxin is highly toxic. Okay, fatal dose is a small amount is already fatal. Endotoxin is low, toxic level is low, so you need hundreds of micrograms of it to cause to be fatal lah. Okay, so endotoxin causes fever and septic shock, but exotoxin have various effects. Okay, in one bacteria can cause diarrhea, one can cause paralysis, right? One can cause a pseudomembrane, okay, in the throat, okay. So here mode of action mainly by production of TNF and IL-1 by the body. Okay, so it's an immune response that is mainly causing the clinical effects, whereas here there's various modes in exotoxin. Antigenicity, high titer antibodies. This is more antigenic compared to endotoxin. Vaccine often used as inactivated into toxoid form and used as vaccine. Here cannot be used in vaccine production. Heat stability, exotoxin is destroyed rapidly at 60 degrees except for staphylococcal enterotoxin. So it can be destroyed with high temperature except for the enterotoxin or exotoxin of staphylococcus. Whereas endotoxin is stable at 100 degrees for up to 1 hour. Okay, so typical diseases caused by exotoxin are tetanus, botulism, diphtheria, okay, pertussis. Endotoxin will be meningococcemia, sepsis by gram-negative rods. Okay? Can and that is, I think, the last slide. Oh no, we still have some. So what is this is just a summary of the infectious process. So bacteria needs to transmit from external source, evade the host, adhere, colonize, and then cause disease symptoms, and then causing specific, non-specific host response, and then it either progresses or resolves. Okay. Typical stages of an infectious disease is the first is the incubation period, followed by the prodrome period, which is the initial symptoms that are seen, specific disease period, and then the recovery period. And some individuals can become chronic carriers, and some infections can develop, can be latent, can be present as latent in their body and then cause infection later. Okay, and that was our last slide.